Hello everyone, welcome back to Microbiome Solutions. Today we're going to learn about the continuation of the biochemical reactions that we have seen in the last class. We will be describing about the assays for the assessment of different metabolic pathways by the bacteria. First one is the carbohydrate fermentation or the OF test, that is oxidation fermentation reaction. So the purpose of this OF test is to determine whether a substrate utilization is an oxidative or a fermentative process for the identification of several different bacteria. So this test is mainly used to identify or to differentiate between two major groups of organisms, that is Enterobacteriaceae, which shows the fermentative property, and Pseudomonas species, which shows the oxidative property. So here we see the utilization of particular substrate to see whether the organism is oxidative or fermentative. So the OF medium consists of high concentration of carbohydrate, that is 1% of carbohydrates, small concentration of peptone. Indicators includes bromocresol purple, which changes the color from purple to yellow, and andrade's acid fission indicator, which will change the color from pale yellow to pink, phenol red, red to yellow, and bromothymol blue, green to yellow. So we commonly use bromothymol blue for the oxidation fermentation reaction, which changes the color of the green colored media to yellow color if it is a fermented. So this is the principle showing glucose oxidative fermentation reaction, where there is carbohydrate is seen in high concentration in the medium. So as you can see in this first picture, one tube is overlaid with mineral oil, which gives an anaerobic condition for the organism to grow, whereas the left tube is not with the mineral oil. So the bacteria will grow aerobically in the tube where mineral oil is not present, and bacteria will grow anaerobically where the tube mineral oil is seen. So both the tubes of OF glucose inoculated with the test organism. So both these tubes will have organism inoculated in it, and after the inoculation of the organism, one tube will be overlaid with the mineral oil to create an anaerobic condition. So these tubes are incubated at 37 degrees Celsius overnight, and if the organism is an oxidizer or a non-fermenter like Pseudomonas, it will give a slight yellow color at the 1/4 of the tube. On top 1/4 of the tube, there will be yellow discoloration or yellow color change if the organism is a oxidizer. So it is oxidizing the sugar which is present in the media, not completely, and the aerobic condition. And whereas the anaerobic tube, there is no change. And you can see the second one, if it is a fermenter like Enterobacteriaceae, both the tubes have changed the color from green to yellow. So it is a complete fermenter of sugars. And the last tube is the non-utilizer, which is neither utilizing the sugar in aerobic condition nor utilizing it in an anaerobic condition. These are the three possible results that you can get after doing a OF test or oxidative fermentative reaction. Either it can be an oxidizer organism or a fermentative organism or a non-utilizing organism. So interpretation, if it is a glucose fermenter, when the acid production is detected on both tubes, since fermentation can occur with or without oxygen. So fermentation is a process where it is happening either in the presence of oxygen or in the absence of oxygen. So if the organism is a fermenter, in the aerobic as well as anaerobic tube, there will be color change. And the organism is a glucose oxidizer, acid is detected by the open aerobic tube, not completely, but upper 1/4 of the tube will be showing yellow color, which denotes the acid production by the organism. And non-utilizer, some bacteria do not use glucose as a substrate for its multiplication or for its growth. So such organism will not show any change in the aerobic as well as in the anaerobic tube. That comes as a non-utilizer. This is the interpretation of OF test. This is the same thing Open tube and closed tube, what is the type of metabolism? And open tube is an acid, yellow, and closed tube if it is an alkaline, showing green, the organism is oxidative, and both the tubes are turning yellow. This is a fermentation reaction. Alkaline and alkaline, that is green and green, it is a non-saccharolytic or a non-utilizer organism, and this oxidative fermentation medium is called as Hugh-Lifson OF medium, that is identified by two scientists, Hugh and Lifson. So examples for the oxidative fermentative organism: fermenter from the Enterobacteriaceae, it is Escherichia coli; oxidizer, it is Pseudomonas aeruginosa; and non-utilizer, Alkaligenus faecalis. Next is the carbohydrate fermentation in triple sugar ion agar or TSI agar. So this is the initial step in the identification of any Enterobacteriaceae, that is fermenter we have identified by using OF test, and then we are seeing whether it is a carbohydrate fermenter in TSI. So the principle is the action of many species of microorganism on a carbohydrate substrate results in the acidification of the medium with or without gas production. An iron salt which is present as ferrous sulfate and ferric ammonium citrate in the medium react with the H2S to produce an insoluble black precipitate. So you can see the presence of hydrogen sulfide and the acidification of the medium with or without gas production due to the fermentation of carbohydrate. So two things can be seen in the triple sugar ion agar medium or TSI medium. It is composed of the protein source like beef extract, peptone, yeast extract, and proteose peptone. Sugars like lactose, sucrose, and glucose. Indicators: phenol red shows the carbohydrate fermentation, ferrous sulfate indicates hydrogen sulfide production, sodium thiosulfate acts as a source of sulfur atoms, and sodium chloride acts as a osmotic stabilizer. These are the components of a TSI medium. And the biochemical reactions, that is carbohydrate fermentation, gas production, H2S formation. These are the different reactions seen in TSI. First, it is a carbohydrate production. If carbohydrate is fermented by the organism, due to acid production, the color of the medium from red to yellow. So if it is a yellow deep, it shows that glucose is fermented. Glucose is having highest percentage in the medium. So if the yellow color is a deep yellow, glucose is fermented, and yellow slant is seen, that can be lactose or sucrose that is fermented. So if it is a glucose fermenter, both the slant and the butt will be changed to yellow color. If only slant is yellow, it can be either a lactose or sucrose fermenter organism. And gas formation is denoted as a bubble formation showing cracking or splitting of the agar, upward displacement of the agar, or pulling away of the medium from the walls of the test tube. This is how you can see the gas formation by the organism. And H2S production is seen by blackening of the butt of the tube, that is ferrous sulfate is produced, which is a black precipitate, gives black color to the butt of the medium. So common reactions, that is acid slant, acid butt, that is yellow by yellow, gas formation but no H2S. So first picture A shows the reaction: acid slant, acid butt, gas formation, no H2S. All sugars are fermented in this reaction with gas formation. Since there is no blackening of the butt, it denotes there is no H2S production. So examples includes Escherichia, Klebsiella, and Enterobacter. Next is alkaline slant, acid butt with H2S production with H2S. So that is red color slant, yellow color butt, gas formation, that is cracking of the medium, and H2S production, blackening of the butt. So the reaction here will be glucose is fermented since the butt has turned yellow. Lactose and sucrose is not fermented since the slant is still red, with gas formation, that is production or the cracking of medium, and black precipitate in the bud. Examples of organisms including Salmonella, Proteus and Citrobacter. Next is alkaline slant, acid bud but no gas, no H2S. That is slight red color slant, yellow color bud, no gas, no crackling, no H2S, no black precipitate. Here glucose is fermented, lactose and sucrose is not fermented. Since the bud is yellow and slant is red, glucose is fermented, lactose or sucrose is not fermented. No gas formation, no black precipitation. Examples include Shigella, Providentia, Serratia and anaerogenic Escherichia coli. Next is alkaline slant, alkaline bud, no gas, no H2S. So it is red color slant, red color bud, no crackling, no black color. So here no sugars are fermented, neither lactose, neither glucose, neither sucrose is fermented, no gas production, no black precipitate. So it clearly says that it is a non-fermenter organisms. Examples include Pseudomonas and Alkaligenes. Next is acid slant, acid bud with gas with H2S. So here the yellow slant, yellow bud, gas is seen and black precipitate is seen. Here all the sugars are fermented with gas formation with black precipitate in the bud. Example is Citrobacter fruendi. So next important reaction for the metabolic pathway assessment is methyl red Vergus perovskite test, that is MR-VP test. This test is done to identify the lactose fermenting enterobacteriaceae such as Escherichia coli, that is MR positive, VP negative, whereas most members of the Klebsiella, Enterobacter, Serratia, Hafnia group which are only VP positive, MR negative. So metabolism of glucose using MR and VP pathway. This is the diagram showing the metabolism of glucose. Glucose is converted to pyruvic acid and acetone. If it is converted to pyruvic acid, because of the mixed acid fermentation, the pH will turn less than 4.4, and if it is turning less than 4.4, it will turn the color to red upon the addition of medium, and this acetone will convert to diacetyl at the presence of atmospheric air and KOH. Upon the addition of naphthol plus creatinine, pink red color complex is formed, that is VP positive. The principle of methyl red test or MR test. The first pathway what we have seen, the mixed acid products, that is lactic, acetic and formic succinic acids result in leading to a decrease in the pH of the medium, that is less than 4.4, which gives a positive methyl red test. So the pH must drop to 4.4 and less than 4.4 for the MR indicate to take on its acidic red color. So if it is dropping 4.4 or even less than that, only the MR reagent or MR indicated will take up the acidic red color. So example for positive MR test is Escherichia coli, negative is Klebsiella pneumoniae. Next is the principle of Vergus perovskite test or VP test. Here in the second pathway, acetyl methyl carbinol or acetone, which is seen as an intermediate product to butylene glycol. So it is the neutral product detected in the VP reaction. So in the presence of atmospheric air and 40% of KOH, acetone is converted to diacetyl form, which results in the red color in the presence of the indicator alpha naphthol. So the example for VP positive is Klebsiella pneumoniae and negative is Escherichia coli. Interpretation of MR and VP test. If MR is positive, there will be distinct red color at the surface of the medium, and if it is negative, yellow color is seen at the surface of the medium. And VP positive, pink red color at the surface of the medium, negative yellow color at the surface of the medium. So the base medium for both these tests is a glucose phosphate broth or GP broth, to which we inoculate the organism, and upon inoculation the organism is grown and the next day or after 18 to 24 hours we take and add the respective reagents to test for MR and VP. Next is the amino acid degradation, decarboxylase and dihydrolase reaction. So the purpose is to determine the production of decarboxylase by the bacteria, that is Enterobacteriaceae. Principle is decarboxylase enzyme which removes the carboxyl group from the amino acid lysine and ornithine, and dihydrolase enzyme removes the carboxyl group from the arginine. So these are the three amino acid used in the reaction, that is lysine, ornithine and arginine. Lysine and ornithine gives decarboxylation reaction and arginine will undergo dihydrolation reaction. So glucose base without the amino acid and tubes containing glucose and amino acids are inoculated, and decarboxylation and dihydrolation are anaerobic reactions. So inoculated tubes are overlaid with mineral oil to exclude air. So you have the amino acid preparations which will be overlaid after the inoculation of the organism, which will be overlaid with the mineral oil to create the anaerobic condition, same like you have seen in the OF tube. The composition of the medium, the decarboxylase medium is called as smallest decarboxylase medium, contains glucose, amino acid substrate, that is 1% lysine, 1% of arginine, 1% of ornithine. So you have to prepare it separately. So for lysine you have to add glucose plus 1% of lysine plus pH indicator, and for arginine 1% of arginine, glucose and indicator, same like for the ornithine. So the indicator are used are bromocresol purple and phenol red. Bromocresol purple at alkaline pH will stay in purple color. When it changes to the acidic pH it will change to yellow color, and the phenol red indicator at alkaline pH it will show the color as red, at acidic pH it will show the color as yellow. So this is the specific amine products. Lysine is converted to cadaverine upon the reaction of the bacteria. Ornithine is converted to putrescine, and arginine is converted to citrulline, and citrulline is further converted to ornithine. Ornithine is further converted to putrescine. So arginine to citrulline conversion is dihydrolase reaction. Citrulline to ornithine reaction is a decarboxylation reaction. So early incubation, both the tubes are yellow due to the acidification of the indicator, that is bromocresol purple, by the acid end products of glucose fermentation. So if amino acid is decarboxylated, alkaline amines are formed and causes the indicator to revert to an alkaline pH. So if amino acid is decarboxylated, alkaline amines are formed and indicator will be changing to alkaline pH, that gives purple color. So interpretation will be control tube will turn as yellow because of the glucose fermentation. Viable organisms and pH of the medium has been lowered sufficient to activate the decarboxylase enzyme. Only at the lower pH decarboxylase enzymes get activated. And positive test shows purple decarboxylation reaction, formation of the alkaline amines from the decarboxylation. So since the alkaline amines are formed, the indicator will be reverted to alkaline pH. So the positive test results will show purple color in the tube. So you can see the molars decarboxylase medium. Positive tubes of decarboxylation shows purple color. Negative decarboxylation shows yellow. That is no decarboxylation, only glucose fermentation is seen because of the yellow color. So first picture what you are seeing here is a control tube without any amino acid, only glucose is fermented, and B is the arginine positive test tube, and C is the lysine negative, only glucose is fermented, and D is ornithine positive reaction. So the decarboxylase dihydrolase reactions of Enterobacter cloacae is represented here. So we see the difference between Enterobacter cloacae and Klebsiella pneumoniae in amino acid decarboxylation dihydrolation reactions. So arginine is positive for Enterobacter cloacae, negative for Klebsiella. Lysine is negative for Enterobacter and positive for Klebsiella. Ornithine is positive for Enterobacter and negative for Klebsiella. These are the basic differences in arginine, lysine and ornithine for Enterobacter and Klebsiella. Next is the deaminase reaction. So deaminase reaction is to determine the deaminase activity using the amino acid phenylalanine or tryptophan. Only Proteus, Providentia, Morganella species possess this deaminase enzyme. So this deamination of the amino acid results in a colored compound with the addition of 10% ferric chloride to the bacterial growth. Here the phenylalanine will get converted to phenyl pyruvic acid, that is PPA, plus 10% of ferric chloride, that is by the enzyme phenylalanine deaminase, which gives green color to the medium. So phenylalanine is converted to phenyl pyruvic acid by phenylalanine deaminase. Next is tryptophan get converted to indole pyruvic acid in the presence of ferric chloride by the enzyme tryptophan deaminase. So the deaminase enzymes are mediating this reaction leading to brown color. Interpretation of phenylalanine deamination test. Positive deamination for phenylalanine shows intense green color and positive deamination for tryptophan shows brown color and negative shows the slant retains its original color after the addition of ferric chloride reagent. So negative for deamination test is Escherichia coli, positive is Proteus vulgaris. And next one is the lysine ion agar. The purpose of lysine ion agar is to determine the ability of the organism to deaminate lysine, decarboxylase lysine and produce hydrogen sulfide, and also to identify organisms like Salmonella, Proteus, Providentia and Morganella. The composition of LIA is proteins, sugar that is glucose, amino acid lysine, sulfur, indicators like ferric ammonium citrate for the detection of hydrogen sulfide production, bromocresol purple for the carbohydrate fermentation reaction. So principle is, as glucose fermentation occurs, the deep or the butt of the tube turns yellow. This is the same principle as like TSI or Kliegler ion agar. So lysine decarboxylation produces alkaline, cadaverine and converts to the reversion of the deep from the yellow to purple. So lysine deamination occurs in the presence of oxygen on the slant and result in the production of a red color. And here hydrogen sulfide is noted by a black precipitate, that is hydrogen sulfide production because of the reagent, and in the deep H2S reacts with ferric ammonium citrate. Interpretation is seen, lysine decarboxylation in the bud, that is positive, purple color is seen in the bud if it is positive and yellow color is seen in the bud if it is negative. And lysine deamination is seen in the slant, positive seen as a red color in the slant and negative seen as a purple color in the slant. So this is the picture showing lysine decarboxylation and deamination reactions. This is about today's lecture. If you have any doubts, please post it in the comment section. Please do like, share and subscribe to our channel. Thank you.